Hello, my name is Elena Vecino. I'm professor of cell biology at the University of the Basque Country, and also I'm the PI of the experimental ophthalmobiology group. I'm going to present the main lines of research of the group during the last years. Let me introduce you my closest collaborators, Dr. Juan Durán de la Colina and Javier Araiz, who started to collaborate with me 25 years ago. And after that, different students and clinicians have been incorporated doing the PhD with us and collaborating in different projects. Right now, the group is formed by 10 doctors and a variable number of PhD students. We are collaborating with the hospitals of Cruces, Araba, and Donostia, and the Eco Institute of Clinical Ophthalmology in Bilbao, and several other hospitals around Spain. We collaborate with several universities that we will point out during the presentation, and abroad universities also. The three main lines of research are the neuroprotection, retina, and glaucoma, which is the core of the group, inflammation, and also another important line is the eye surface. Let me introduce you what we are doing with this neuroprotection, retina, and glaucoma line. Glaucoma is a neurodegenerative disease that affects 4% of the population and is caused by increase of intraocular pressure. But at the end, the retinal ganglion cells, which are connecting the eye with the brain, are those who died. There are very important Miller cells, which are the glial cells in the retina that are protecting the ganglion cells, and we are working also with those. In 2004, the American Glaucoma Foundation supported us with a prize to develop a model of glaucoma in mini pig, something that we did. At that time, Dr. Marta Galdós and Carlos Micó did the PhD with us, developing the model of glaucoma in mini pig, but also analyzing the effects of the glaucoma by OCT, electroretinogram, and electron microscopy. We set up the model that is used for different aspects all over the world. More or less around that time, Dr. Aritz Urkola, which is the head of glaucoma at the Txagorritxu Hospital in Vitoria, did a PhD with us. He developed, in addition to the episcleral vein cauterization model, who was developed by Dr. Sharma in New York. As I say, he developed the BEATS model of glaucoma, also called microspheres. Is a model used all over the world in mice mainly, and he compared these different models in terms of how many cells die and how were the impact of the pre-trabecular or post-trabecular model of glaucoma. Dr. Aritz Urkola at present has his own line of research studying the biophysics and lipidomics of the aqueous humor of patients with glaucoma and other diseases, in addition to the study of electron microscopy of the Slane channel. Both projects are in collaboration with our group. Moreover, Iñaki Rodríguez Aguirreche did the PhD also with us. He is now the head of glaucoma at the Hospital Donostia in San Sebastian. He was looking more at the scar and how using different biomaterials could avoid the formation of the scar after the trabeculectomy operation. Our expertise in glaucoma have led us to collaborate with different companies and pharmaceutical companies to test different compounds in the decreasing of the intraocular pressure. And very recently, together with the company Silentis, we have a Retos Mineco grant to develop nanoparticles that could protect the interference RNA direct to load the intraocular pressure. We have been collaborating with Dr. María José Alonso with her group in Santiago de Compostela, expert in nanoparticles. Now these new compounds are in a phase 3 clinical trial, direct to the young population and also older population. But as I said, together with the models of glaucoma, we are experts in cultivating culturing Miller cells and retinal ganglion cells, and to look at the interactions between both. Doctora Noelia Ruzafa and Sandra Pereiro did the PhD with us, and they are leader in this aspect of the group. We have looked at the proteomics of the secreted factors of the Miller cells and also the lipidome of both type of cells and the interactions between them. Now Sandra Pereiro is doing the postdoc in Coimbra and looking at the effect of the pressure in this type of interactions. Another line of research is the inflammation. When Dr. Alex Fonollosa was incorporated to the Basque Country, contact us, he was interested in study the effect of somatostatin in the inflammation and edema. We developed a system to test the effect and how to restore the equilibrium in the retina. Moreover, hyperrefractive dots that appears in inflammation in the retina, we were analyzing in vitro, and that hyperrefractive dots correspond to microglia, and we did a study on that. Dr. Javier Araiz, one of our closer collaboration from the beginning of the group, is interested in the P-retinal membranes. He is an expert in retina, and we are analyzing thanks to a project with the Basque government, funded by the Basque government, to look at the proteomics and lipidomics of these epiretinal membranes. Another important line of research in our group is the eye surface. Doctora Arantxa Cera and Dr. Durán de la Colina are the responsible for this line, and at present we have a Retos Mineco project to identify the lipidomics of the tears. During the last 15 years, we have been studying the proteome of the tears, and we have published more than 20 papers on this field. However, the title of the talk was From the Tears to the Brain, and at present we have an important project in relation to the biomarkers to diagnose the Parkinson. We are collaborating with different groups of neurology and ophthalmology and Tecnalia, which is an important company in the Basque Country, to design a point of care to identify the early diagnosis of Parkinson. As summary, from the last 25 years, our group have supervised 32 theses and published more than 100 papers, 47 during the last 5 years. The group is very active and collaborative with different labs from Spain and different countries, and open to new projects. And one of the projects during the last 2 years, it has been to study the eye of the largest mammal in the planet, the whale. A very interesting project, and the results in pictures have been exhibited for the moment in Bilbao and few other cities in Spain, and the exhibition has been completely adapted to blind people and people with low vision. And it's a project for education. But this will be another talk. I want to finish this presentation with a picture at electron microscopy level of the whale's retina. This was part of the exhibition. Thank you very much for your attention.